Hello, and thanks for joining us today for the latest Technology Networks webinar, Cloning in the Third Dimension, Breakthroughs in 3D Biology. I'm Kate Harrison, Science Writer for Technology Networks, and I'm here to moderate the event. I'm very pleased to have Dr. Allysa Stern and Dr. Scott Magness joining us today as your presenters. A warm welcome to both of you. Dr. Magness is an Associate Professor at the UNC-NCSU Joint Departments of Biomedical Engineering. His research is focused on investigating the genetic mechanisms that control the behavior of intestinal and colonic stem cells in health, injury, and disease states, including inflammatory bowel disease and cancer. Dr. Stern obtained a PhD and Master of Science in Physiology from North Carolina State University. Her background is in comparative physiology, with a concentration in cell biology and developing 2D and 3D in vitro models for drug and toxicological studies. Following the webinar, we will have a Q&A session, and we welcome any questions you may have. You can submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the right-hand side of your screen and click Send, and we will answer as many as possible during the time available. For any questions we don't have time to get to, we'll make sure you're contacted offline with an answer. If you experience any technical issues, please click the chat icon in the bottom right corner to request support. Please remember, you can ask questions at any time, even while watching the webinar on demand. So, without further ado, I will now hand over to Allysa and Scott.
Thank you, Kate, and thanks everybody for joining us for the webinar today. I'm really excited to talk to you about our cloning in the third dimension and some of the breakthroughs that we're making in 3D cell biology. In today's webinar, I'll demonstrate a novel workflow for organoid culture that enables both clonal organoid development and monitoring iPSC differentiation into organoids. I'll also describe how you can generate custom 96 well plates with single organoids of interest for your downstream assays and assessment. And Dr. Scott Magness will discuss his new approaches to investigate tumor cell heterogeneity using single organoid transcriptomics. Induced pluripotent stem cells have sparked widespread enthusiasm for the development of new models of human disease. This is partly because they replace the previously used embryonic stem cells, and also because they provide the potential to study both disease and patient-specific aberrations. These oncogenic or diseased iPSCs can be derived in several ways, either directly from patients with known diseases or mutations, or by performing gene editing using normal, healthy donor iPSCs. And while the scientific community recognizes the potential of being able to differentiate these iPSCs into virtually any cell type of the body, it's really the combination of the iPSC and more recent organoid technology that allows us to utilize a more physiologically relevant, tissue-specific in vitro model to study things like disease development and progression, new target discovery, as well as drug screening downstream. The data I present today will focus on cerebral organoids. The development and commercialization of the cerebral organoid technology has really given researchers access to a physiologically relevant in vitro human model for critical developmental biology and cancer research. So it's no surprise here to see this exponential increase in the utility of these cerebral organoid models in the literature over the last decade, specifically for things like glioblastoma, Alzheimer's disease, and stroke. However, despite the promising applications of using these models, these workflows to generate the organoids are really hampered by challenges of scalability, reproducibility. The workflows are very manually intensive, and altogether, this really limits them to low to mid throughput applications. So while as scientists, we recognize the potential of moving from these diseased iPSCs into the organoid models, the reality is the road to get there looks a little bit more like this. So despite the fact that the methodologies for generating the organoids is really incredibly useful, we need technologies that overcome some of the challenges to really accelerate them into preclinical applications.
Okay, so poll question 1. What is the biggest pain point in 3D cell culture? low throughput, manual workflows, lack of reproducibility, or you're not growing organoids, but you do want to? Can you please submit your answers to the poll now?
Now that we've discussed some of the challenges in these organoid workflows, the big question is, how do we scale organoid culture methods for cell-based assays? Today, I'm going to introduce you to a technology that addresses many of the challenges of organoid culture and screening. The core of our technology is the cell-wrapped array. This is an innovative culture plate. Cells are seeded into the cell-wrapped array, much like they would be in any other tissue culture consumable. Importantly, these cells share a contiguous media volume, just like they do in bulk culture. However, within the reservoir of the cell-wrapped array, we fabricate an elastomeric microwell array. This provides spatial segregation of thousands to tens of thousands of cells in one reservoir. Importantly, at the bottom of each of those microwells is a microscale polystyrene growth surface we call the cell raft. These cell rafts are releasable from the cell-wrapped array. Just to orient you a little further, here you can see an animation of the cross-section. So in white, we have this elastomeric microwell. At the bottom of the microwell is this magnetic polystyrene cell raft. Cells attach and adhere to the cell raft just like they would any other tissue culture surface. But as I mentioned, one of the key features of the cell-wrapped array is that while those cells remain spatially segregated, they're sharing that contiguous media volume, allowing them to exchange growth factors, which really improves the viability and the growth of the cells on the array. I've just introduced you to the cell-wrapped array that provides flask-like culture conditions and single-cell separation. We also use image-based software-guided selection to find your cells or colonies of interest, and then isolate those cell rafts containing your cells of interest using the cell-wrapped air system. While this technology was originally developed for two-dimensional applications, we found all of these key features of the cell-wrapped technology to be uniquely suited to address many of the challenges in organoid workflows. Using the cell-wrapped technology, we're culturing organoids using a different approach that I like to call outside the dome. Here in this image, you're looking at a stitched together images of the entire array in bright field. In green, each of those circles highlights an individual organoid grown on a cell raft. So you can see that we're able to grow and maintain hundreds of individual organoids in a single focal plane. Because of the cell-wrapped array, we can serially image each of those organoids over time. And we can use those images to perform phenotypic characterization of the population to identify individual organoids of interest for our downstream applications. The workflow for generating iPSC-derived organoids is really straightforward. It starts with preparing the 3D cell-wrapped array. This is our largest format with 500 by 500 microwells and cell rafts. We dissociate our iPSCs into single-cell suspension, seed those iPSCs in a dilute extracellular matrix onto the cell-wrapped array. After those cells and ECM have settled into the microwells and polymerized, we begin performing two-dimensional scans of the array to track organoid growth. Here in this top panel, our little white arrow is pointing to a single iPSC, and you can see over time as that iPSC develops into an organoid. To find the cell rafts that contain your organoids of interest, we use cell raft cytometry. This uses advanced bright field analysis with the option of incorporating 3 fluorescent channels. Users can define populations to find the cells that they're interested in. So from a clonal organoid perspective, this might look like single iPSCs at time 1 and organoids at a later time point. Importantly, the subset feature allows you to overlay any of those populations, giving you a temporal analysis of each individual cell raft. So here in this top panel, you can see I have a population for single cells in A and organoids in B. By overlaying those populations, I'm getting a list of cell rafts, and I can rapidly map those cell rafts for isolation. There are three key pieces that enable organoid isolation. The first is the cell raft array, which I've already introduced you to. We have a release needle that's mounted on the objective that penetrates the elastomeric floor of the array, releasing the polystyrene cell raft from its microwell. And then because those polystyrene cell rafts are doped with magnetic nanoparticles, we use a magnetic wand to dip into the reservoir and transfer those cell rafts to the 96 well plate. Importantly, this process is really very gentle. There's no enzymatic dissociation, there's no fluidics. The organoids remain fully intact and attached to the cell raft throughout the process as they're transferred into the 96 well plate. This animated video is going to walk through the process of cell raft isolation. There you can see, in this case, it's a single green cell. The needle releases that cell raft from the microwell. The magnetic wand picks up the cell raft and transfers it to the 96 well plate at our designated location. While I really like that the animated video orients you to the process, really the live view image of this is quite striking. So here on the left, again, this is our 500 micron cell raft array. So you're seeing an organoid there that's roughly 300 to 400 microns in diameter. As I play the video, you'll see the focal plane adjust as the release needle releases that cell raft from the microwell. The wand dips into the reservoir and then picks up the cell raft and organoid attached to transfer it into the 96 well plate. So there you can see the wand moving over to the mapped location on the 96 well plate. Now that I've introduced you to the technology, I'd like to show you how we're using this technology for iPSC organoid culture and isolation. In the beginning of the webinar, I briefly discussed the challenges of these workflows. So this is the standard workflow for generating cerebral or choreplexus iPSCs. You can see here we start by taking our iPSCs, seeding them in bulk into a 96 well plate for embroid body formation. You would then pipette each of those embroid bodies into a 24 well plate for neural induction. After neural induction, you would embed each individual organoid in a droplet of matrigel. And then after the matrigel has polymerized, you would wash those matrigel droplets into a 6 well plate for expansion and differentiation. So you can see here that this process is very manual, would require expert users to be efficient at. And in addition to that, throughout the process of differentiation, as you're moving organoids from plate to plate, you lose track of any ability to monitor each organoid's development over time. We wanted to see if we could take this multi-step process and collapse it onto the cell raft array. So for our workflow, we take that same bulk iPSC suspension. We seed those single cells into the cell raft array and embroid body formation media with an extracellular matrix suspension. After that ECM has polymerized, we begin performing our two-dimensional scans. And for neural induction and expansion, we're simply performing media changes in the reservoir of the array. For our cerebral workflows at day 10 after expansion, we're selecting organoids to isolate into a 96 well plate to do single organoid maturation off of the array. In addition to a simpler workflow, we've also reduced reagent usage for this workflow. So here you're looking at the reagent and media volume for that standard cerebral organoid workflow. With the cell raft array, you can see here we're using significantly fewer cells. And for most of the steps of the differentiation protocol, we're also using less than a half of the media required and a third of the matrigel required to form those organoids. Importantly, with this cerebral organoid workflow, ultimately what you end up with is a 6-well plate with dozens of organoids in each well, with a maximum of 96 organoids. Whereas with the cell raft array workflow, we're generating hundreds of organoids on the cell raft array, and then we're able to transfer them to the 96 well plate so that we have a full 96 well plate of single organoids for our downstream use. One of the most valuable features of the cell raft technology that increases the reproducibility of your downstream assays is the time course imaging of each organoid. So here on the left, you can see this is a field of view image of a heterogeneous population of organoids. For these organoid workflows, we intentionally co-cultured 2 different iPSC lines, one with a green fluorescent reporter and one with a red fluorescent reporter, to demonstrate the heterogeneity of the organoid population. So here you can see we have green organoids, we have red organoids, and we even have some that are dual fluorescent. The beauty of the cell raft technology is you're able to parse out each of these phenotypes and look at the organoids on a cell raft to cell raft basis. So here we have a single cell derived red organoid, we have a green organoid, and then this dual fluorescent organoid actually formed from a single red and single green cell. With these images, you can easily imagine the universe of opportunity you have now to tailor your downstream organoid applications by assessing a heterogeneous population, finding the particular phenotypes you're interested in, and performing your downstream assay on specific single organoids. We've performed this workflow with several iPSC differentiation protocols. Here you're looking at choreplexus organoids. For this experiment, we used the array to monitor differentiation over time. So here you can see a red iPSC as it goes through embroid body formation, neural induction, expansion, and then the first stage of choreplexus differentiation. For this particular experiment, we were looking at the differentiation over time of individual organoids to interrogate the heterogeneous population of organoids. So essentially using the cell raft array as like a cell-based assay to monitor the differentiation across the heterogeneous population. For the remainder of today's talk, I'll be focusing on the cerebral organoids. For this particular workflow, we performed embroid body formation, neural induction, and expansion on the array out to day 10. As I mentioned earlier, the great thing is that cell raft cytometry helps you find cell rafts with your organoids of interest. So for this particular experiment, we were interested in clonal organoids. So we set up a population for finding single iPSCs at time 1, and then organoids of 100 to 300 microns in diameter at time day 10. And as I mentioned earlier, by overlaying these 2 subpopulations, we now have a list of organoids that were single cell derived for our downstream application. In addition to giving you a list of cell rafts for isolation, cell raft cytometry provides an opportunity to quantitate these differentiation protocols. So you can see that we've done this protocol on an N of 3 arrays per differentiation workflow. On average, for each array, for the cerebral workflow, we're generating over 225 cell rafts that contain single organoids, and roughly 275 for choreplexus. And of those over 200 single cell or single organoid cell rafts, more than 50 of those were derived from single iPSCs. This gives us a single cell organoid forming efficiency of roughly 17%, which is quite high, comparatively speaking, for many clonal organoid workflows. The cell raft array, as I mentioned before, also allows you to use significantly less media and reagents to generate these hundreds of organoids compared to the standard workflow. So you can imagine that by seeding multiple arrays, you're able to exponentially increase the total number of organoids for your downstream applications. After we selected those day 10 cerebral organoids, we isolated them directly into maturation media into the 96 well plate. Here is this particular organoid at day 10 immediately after maturation. And you can see several images out to day 37 as this organoid continues to mature in the 96 well plate. Because we're maturing these in the 96 well plate, we can continue to monitor each individual organoid throughout that maturation process, which would not be possible using the standard methodology. In addition, you can see here that not only does the organoid body continue to grow, but because these organoids are not in bulk suspension, we get this really beautiful neurite outgrowth. And by day 37, we have these advanced neural networks coming off of our organoid body. This allows us to study things, study neural outgrowth from the organoids, which would not be possible using the suspension culture method. While the continued growth of that organoid and the neural outgrowth was noted, we wanted to perform a visual confirmation of neural differentiation by staining these organoids about 2/3 of the way through their maturation. So to do this, we took those non-green fluorescent organoids. So here you can see this organoid at day 10 on the array is not GFP positive. On day 28, we stained those organoids with NeuroFluor NuO. This is a membrane permeable dye that stains live neurons green. You can see that we have not only this brightly glowing organoid body here, but each of these neural projections is also staining green, indicating that we have live neurons growing off of the organoid. At this point in the experiment, I'm on my way to having a full 96 well plate of mature cerebral organoids. And really, there's endless opportunities as to the applications that I could have used these organoids for. I come from a toxicology background, so I wanted to perform a quick toxicity screening using mature cerebral organoids. As many of you may know, alcohol is a neurotoxicant. So my hypothesis was that an acute alcohol exposure to these mature cerebral organoids would induce apoptosis. So on day 42, after those organoids were fully mature, I treated 6 replicates with a 5-point dose curve of ethanol at known non-toxic and toxic concentrations. And then to measure the induction of apoptosis, I used the caspase-3,7 glow assay to measure caspase activation after that 6-hour exposure. These images were taken 2 days prior to the ethanol exposure. So this is pre-treatment. What I wanted to point out here is that you're looking at 3 of 6 replicate wells across my dose curve here. If you recall, at day 10, while these organoids were on the array, I selected them for a diameter of 100 to 300 microns. And by doing so, what I've enabled is a downstream, a 30-day downstream, fully mature cerebral organoid assay, where although these organoids have different morphologies, the size of these organoids is roughly the same. So for some of the endpoints that are specific on cell number, this is a very key feature of the CellRaft technology. Here for that caspase activation assay, you can see I get a nice dose-dependent increase in the caspase activity relative to the control with apoptosis induction at these high doses of ethanol exposure. So altogether, what I've demonstrated for you today is that the CellRaft technology really allows you to create these custom 96 well plates with your organoids of interest for whatever your research goals are. I showed you a toxicology approach today, but these single organoids can be used for transcriptomics, drug screening, as well as looking at a heterogeneous patient-derived population on the array. To wrap up here, I've shown you today that with the cell-raft array, you can grow hundreds of spatially segregated organoids in a single focal plane. You can reliably track each of those organoids' growth over time and easily isolate single organoids that you're interested in for your downstream use. I've also shown you that we've been able to simplify some very complex workflows and generate reproducible organoid assays with confidence using the cell-raft technology. Before I close here, I'll just leave you with this last sort of thought-provoking question, which is, how would a full plate of characterized single organoids enable your breakthrough research?
Okay, poll question 2. Do you want to learn how to grow a full plate of characterized single organoids? Yes or no?
And with that, I'll just quickly acknowledge some of my cell microsystems colleagues who contributed to the development and some of the data you saw here today. And I'll thank you for your time and turn the floor over to Dr. Scott Magness for his portion of the webinar.
So thank you, Allysa, for inviting me to share some of our work with you. My name is Scott Magness, and I'm an associate professor at the University of North Carolina at Chapel Hill. And my primary research focus is on GI intestinal stem cells, so from the intestine, stomach, and colon. And we really try to understand the very basic genetics of how stem cells operate. So what basically keeps them as stem cells or allows them to differentiate into the different postmitotic lineages, otherwise known as stemness. And then how that these genetics regulate the cell biology, mainly the tissue regeneration of the gut. The gut is the basically one of the most regenerative tissues you have in your body. And so it's a stem cell driven process. And when that goes wrong, you have a lot of things that can happen that are associated with health conditions like inflammatory bowel disease and cancer. And we hope to understand all of these processes to help us develop new cell-based therapies, ex vivo models of the gut. And we have a number of programs that are associated with development of gut-on-chip technology. One of the things I'm going to talk about today is kind of the transition of our work into single cell biology, really taking a low resolution historical approach and converting it into a high resolution single cell biology approach. So why should we study biology at the single cell level? Most of you can probably answer this already, but emerging studies are indicating that there's really an unappreciated level of cell heterogeneity. We, historically have been studying tissues and organs or bulk conglomerations of cells, and not at the single cell level, which is where biology really happens. Not only does biology happen there are a lot of diseases that are initiated at the single cell level. And by the time we actually know that we have some sort of health condition, those cells typically have grown to the point, at least in cancer, to the point that there is a health condition that needs to be addressed. So again, we go from a low resolution bulk analysis sort of historical approach to studying cells. And what we want to do is to go to a high resolution single cell analysis approach. So what I'm going to talk about first is how we study single cell biology in the normal context, in the physiologic context. And again, my primary research focus is the intestinal stem cell. And what you're looking at here on the left-hand side is a cross-section of the gut. I'm interested in just the lining of the gut. And if you blow up the internal surface of this on the right-hand side is the microanatomical structure of the epithelium of the intestine, small intestine in particular. And at the very bottom, you'll notice that these crypt structures, and at the very base of those, you'll see these red cells in the schematic. Those are stem cells. There's about 6 to 8 of those per crypt. And when they divide, they give rise to this pink transit amplifying population. These are not stem cells anymore typically, and they are being influenced by their environment to become one of the postmitotic lineages that are represented in the villus structures, mostly the gray, green, and blue cells you see here. There's a traditional or a stereotypical migration that happens from the stem cell zone up this axis. You can see by these arrows. And at the very tip of the villus, the cells are sloughed. And this whole process from the stem cell dividing and giving rise to the transit amplifying and differentiating is only about 7 days. So it's pretty amazing that your entire gut lining turns over every single week. And again, when this process goes wrong, you can have disease conditions that are associated with that. What I'm showing here on the right are just the post-mitotic lineages that are housed in the villus structures. So the majority of these are the absorptive enterocytes, and the minority are secretory lineages. And there's about 5 to 6 of those that do different jobs in the gut. They make hormones, they make mucus, they sense microbial taxa inside of the lumen of the gut. And so they have a variety of functions. These are all derived from the stem cell. And really, just in the past 5 to 6 years, we've started to understand how the stem cells sort these different lineages as they differentiate up this axis. But it's still challenging to study that. And really the reason is because we really don't understand how these stem cells function from the stem cell to the transit amplifying zone, and what genes are involved in sorting these different post-mitotic lineages. One of the reasons that we're kind of late in studying this process is because historically there was no way to identify, isolate, and culture intestinal stem cells. And about maybe 10 to 12 years ago, new markers in transgenic mouse models came out. And one that we started looking at was the Soxine EGFP mouse model. You can see here in this green central image here, the base of the crypt, there's green cells that are marked by the transcription factor SOX9. On the right-hand side is just the endogenous gene. And what you'll notice, if you look really carefully, that there are different levels of these, the SOX9 GFP and also the endogenous gene. And initially, we had no idea. We thought that they all might be stem cells, but we didn't understand why they were making different levels of these genes, what might be the difference in their function based on the different levels of the expression of this particular gene. And of course, these transgenic mouse models that express a fluorescent protein are very useful because you can FACS isolate these cells to single cells and then study them. You can look at their gene expression, you can look at their function. And so that's what we did. And so in the center panel here is just the post-sort of Soxine GFP mouse, indicating these different levels. And if you looked at the univariate histogram here on the right-hand side, you can see there are natural peaks that correlate to these different expression levels of SOX9. But we still didn't know which of these were stem cells and which were not stem cells just based on this fluorescent marker. They were all based in the stem cell zone. They're all housed there, but we didn't know what they were based on the markers at the time. Since then, we've published a number of papers that have used transcriptional analysis to identify that the negative cells are mostly the absorptive goblet and Panna cells. The majority of the cells that are the lowest level of green are these transit amplifying progenitors. There's an intermediate level of GFP that are an active stem cell, meaning that they're actively dividing about once every 24 hours. And then there's this very rare population that corresponds to the highest level of GFP you can see on the right-hand side there. And it turns out that these have a variety of functions. There's a heterogeneous population, we think, that exists in this high population that are reserve stem cells, meaning that they don't divide, but they are elicited to reactivate upon some sort of gut injury to generate an active stem cell. So it's a reserve population of stem cells. And then there's hormone-producing cells and also a cell that is a sentinel for worm infections of your gut. And so you can see there's, based on this GFP expression in the very base of the crypt where the stem cells are, there's a variety of functions of these cells. When we took each of these cells and we put them into newly defined culture conditions, what we found was the only ones that would appreciably generate an organoid were the SOX9 low cells. And you can see we placed these into a conventional matrigel culture system. We followed these and tracked these over days, basically through about 10 days. And you can see that the single SOX9 low GFP cell on the left-hand side would transition and generate a fully developed organoid, which you can see on the bottom right. And this is, again, thousands of cells that are generated from a clonal stem cell population. These culture conditions were essential to generate these organoids. And basically it's an ECM, we use Matrigel, and you have to use small molecules or native proteins to elicit a Wnt signal, also a Notch signal, and you have to use proteins or small molecules to turn off BMP signaling, and also a type of apoptosis called anoikis. But interestingly, only about 5% of the stem cells would grow. And so that was interesting. That says that not only does this population ubiquitously express SOX9 GFP low, but only 5% grow. And that indicates there's a functional difference in this population. But we had no real way of evaluating which cells would grow and which ones didn't. And the reason is because these stem cells grow in a three-dimensional context. And so the ability to identify, track, follow, quantify those in 3 dimensions was challenging because the microscopy would not allow it. And so we turned to the cell raft arrays from Cell Microsystems. Alyssa's really gone over this nicely, showing you how the technology works. But what I want to point out here is that the important thing for us was being able to put single cells on the same Z-plane in a high throughput manner and also in a small footprint, because the reagents are very expensive to do in any sort of large scale system. We were also able to track these because of addresses that were on the bottoms of these rafts that were, again, as Allysa pointed out, retrievable. So anything we put on these cell raft arrays, we could retrieve and follow later through gene expression analysis, protein expression, or even growth analysis. And so this is what we did is we plated those SOX9 eGFP low cells in each of the rafts. Allysa went over how those rafts are retrieved and then collected into a 96-well plate. And at the time, this is 2015, we published this paper in Nature Cell Biology. We had to do this manually. And so this is a very slow way. The new air system is amazingly faster, and we can pretty much do this with hands off now and collect 96-well plates very easily. We identified single cells and we traced those over time. And if they generated an organoid, we were able to measure that organoid and then collect those organoids. And what you're looking at here is a gene expression analysis. This is before RNA sequencing. So we had to use high throughput qPCR to analyze the genes that were expressed at each of these different stages of organoid ontogeny, let's call it. And so on the left-hand side, each of these columns in the heat map are an individual organoid or single cell. And you can see at time point 0, the highest activity of the active stem cell signature is in the 0 time point. Because we isolated these based on a marker that was associated with an active stem cell. And then the reserve stem cell, secretory progenitor markers in differentiated cells, the genes are on the y-axis here. You can see that those are lowly expressed. But importantly, it's very heterogeneous. You can see that some of these cells that were identified based on their stem cell signature, at least based on SOX9 GFP low, also had other genes in a heterogeneous fashion that were expressed. And then over time, you can see that there's a change in that gene expression pattern over the ontogeny or growth of this. And by 240 hours, you have a fully developed organoid here on the top right-hand panel. And you can see in each of the columns that not all of the genes are expressed equivalently. So there's got to be some sort of differential or stochastic development in gene expression pattern throughout this process that is leading to an organoid, but even those organoids are not homogeneous. They have heterogeneous cell populations that are likely leading to this gene expression pattern. And so what we found, and just you can go look at the paper for the details, but one of the findings was that some of these stem cells, as they were developing, had differential Wnt responsiveness. And as you know, Wnt is a key molecule and signal pathway that keeps stem cells as stem cells or allows them to differentiate into different cell types. So I'm going to transition now. We studied a lot of normal biology there. We've gone back just to let you know, and we're now doing this on the air system, isolating organoids from normal gut or isolating stem cells from normal gut, generating organoids, and following this using RNA sequencing so we can have a more comprehensive gene expression signature for this ontogeny of organoids. The next thing we wanted to do is to investigate key transition states from cancer at the clonal organoid level. And so now we're looking more at a disease state, or looking at clonal events and disease states. So as most of you know, cancer typically starts probably from a mutation in a single cell. And then over time, those cells can be mutated even further, that eventually may lead to some sort of very aggressive cancer type. And so we partnered with Jim Goldenring and Eun-Young Choi at Vanderbilt University to really study this process of gastric cancer development. What I'm showing you here in this panel here is a normal gland from the stomach, and these are in general very similar to the intestinal crypts, where they have a stem cell population. It's not in the very base of the crypt, it's more in the mid region here on the left. And what happens over time, if you have infection, if you have chronic inflammation, is that the cells will undergo a metaplastic conversion. And then if it continues, you can have a progression into dysplasia and cancer. And so what we wanted to do is to study this far right-hand side at the single cell level to ask the question, are all of these cells that are metaplastic, do they have the same capacity to go to dysplasia and perhaps cancer? And so, if you study this at the bulk level, you lose a lot of the resolution, a lot of the things that might be going on at the single cell level that are important for this particular progression to cancer. And so dysplasia is considered a key transition state between precancer and cancer and gastric carcinogenesis. But the cellular phenotype and heterogeneity and mechanisms of this dysplastic progression have not been elucidated. And so we again turn to the cell raft arrays and the error system to start to develop methods that we can look at the transcriptomic state at the single organoid level. Importantly, though, from a clonal cell that generates an organoid. And so what I'm showing you here is a cell raft array that had been seeded with single cells. And we found this one tract just by chance that had a single cell in each one of these rafts. But what you can see is that that single cell behaved completely differently in these 4 cases, where on the left-hand side, the single cell had a slow growth, and the cells, as they progressed to the right, had much different growth dynamics. But the question is why? Those cells were completely homogeneous based on their isolation strategy. But when we plated them on the arrays and looked to see what they would grow into, they had completely different proliferation rates. And so here's another example. We took these cells from human tumors, gastric tumors, and we plated these in the arrays. And what we found, we looked at the different types of phenotypes that would grow from single cells. And you can see on the top left here, we have type 1 through type 5. Some cells persisted on type 1, but they did not grow. Type 2 is that they attached and grew into these kind of flat structures on the bottom. Type 3 would grow into 2 or 3 cells and then kind of like were stable. They did not die. They just kind of remained stable. Type 4 and 5 grew to full organoids with a lumen. But what we noticed was that there were differences in the cell morphology in the organoid monolayer. So on the left-hand side here, you can see there's a cystic, which is a thin monolayer, and on the right-hand side was more of a columnar monolayer. And so we asked the question, how can we actually study what might be going on at the transcriptomic or gene expression level that would lead to this? And we really had to develop new technologies to study this and develop methods to do RNA sequencing. And so what we did here is we took these organoids, we dissociated them to single cells, and we plated them on the CRAs, cell raft arrays, and then we let them grow over about 11-day period of time until they had a full lumen and were able to be identified by the error system. Then we isolated a number of these, and we had to figure out a way to isolate RNA and develop or generate sequencing libraries from these very small amounts. And we use the Advana system from Fluidigm, which is a microfluidic automated system that allows very low input of RNA. And so what we did is we used the air system to isolate single organoids of different sizes. So on the left-hand side here, what you're looking at is the diameter of the organoid and then the total amount of RNA that we collected from these. So again, we performed this experiment to understand if we could look at the organoid on the arrays and predict how much RNA we could get from these. And you can see that as the organoid gets bigger, as you might expect, that you can achieve a higher yield of RNA isolation. And we identified that this red dotted line here was kind of the threshold at which we could generate enough RNA that would go into the microfluidic library preparation system. And you can see in all of these cases, no matter what the size, that we had a very high RIN of greater than 8 for all of these. So we had excellent quality RNA and quantity from organoids that were generally over about 1,215 microns. And then we sequenced those. And so the question was, if we sequence a single organoid, how close would it be? Would we be capturing all of the genes that would be representative of the entire population? And so we took bulk RNA that was isolated from the same organoids, and we just diluted it. And we looked at individual organoids. You can see here 9 in the black bar, the first data point here on the left. And you can see that we got about the same number of reads in the single organoids compared to each of these dilutions. And on the right-hand side was the ability to align those genes to the genome. And you can see that we were able to align single organoid RNA sequencing data equally well as we could from the bulk RNA. And we found that there was a threshold of about 100 picograms that was optimal for applying to the Advanta system to make the libraries. So you can go down to very small amounts of RNA from single organoids and generate libraries that had very robust data sets. And so then what we did is we wanted to collect more of these. And as you can see here, we collected a 96-well plate. I think Allysa showed these data in her presentation, and we were able to go and visually identify that there was indeed a capture event of these gastric organoids, and we got about 70% isolation efficiency from this particular experiment. So then after we sequenced a number of these organoids, what we wanted to do is really focus first on the columnar and cystic organoid morphologies and ask the question, what are the transcriptomic differences of those? We first thought that perhaps we would be able to see just based on the morphology that the organoids would cluster in either a columnar population or a cystic population based on the genes that are expressed. And so we used Leiden clustering to be able to reveal transcriptomic differences in these organoids. But what you can see is there's really kind of random distribution. There's no real clear population clusters based on morphology alone. But then when we looked at all of the genes that were expressed, we found that there were 2 clusters. There was a cluster, what we call cluster 1 and cluster 2. And again, these did not correlate to the morphology of the monolayer of these particular organoids. So the genes that are driving that morphology are not strong enough to cluster by themselves. So we asked the question, what are the genes in these that are causing this clustering? And we turned to a paper again from our collaborators at Vanderbilt, Eun-Young Choi and Jim Goldenring. We had a curated gene set list of precancerous genes, and we applied this gene set list to each of these clusters. And what we found is there were striking differences in the precancerous gene signatures. Cluster 1 had many more of these genes that were expressed compared to cluster 2, indicating that the clonal organoids that were derived from a perceivingly homogeneous population were indeed not homogeneous, they're heterogeneous, and some seem to be more cancerous than others. So what we're doing now is we're using this information and asking the question, can we take these organoids, these single organoids, and treat them with anti-cancer drugs based on the knowledge of what genes might be conferring some sort of resistance to chemotherapies, and then asking the question, which ones will escape or survive these different chemotherapeutic interventions and allow an organoid to grow. And of course, you can imagine this is very important for drug discovery and development of different cancer therapeutics. And what we've already found in 2 different clusters is that there are genes that are responsible for treatment resistance. And we don't know if they're actually causing this yet, but there are genes associated with different clonal organoid types that we think might be expressing different gene sets that make them potentially resistant to some of the existing cancer treatments and also emerging cancer treatments. So the conclusions, what I've shown you is that clonal organoids show distinct phenotypic and transcriptomic differences. Single organoids and transcriptomic studies can describe and reveal heterogeneity in cancerous gastric epithelium, and that there are 2 populations of cells from human gastric dysplastic tissues, one that has high enrichment for precancerous genes and one that has a lower. And so again, we're continuing, these data are fresh off the press, and so we're continuing to analyze these to better understand these transitions of progression from metaplasia, dysplasia, and full-blown cancer. And so what I'd like to do is to, of course, thank my lab. Most of this work was done by Jared Blight in my lab, who's now a postdoc, and of course, the collaborators at Vanderbilt, Eun-Young Choi and Jim Goldenring. And with that, I'd be happy to try to answer any questions you might have.
Fantastic. Thank you very much to Allysa and Scott there. I'm sure everyone will agree that there were some excellent points raised during that presentation. We will now begin the Q&A session. So to submit a question to our speakers, you should enter your question into the box on the right-hand side of your screen and click Send. We will answer as many questions as possible during the time available. We have actually already had several questions submitted, so I will move straight on to those. But if you still have a question, please, it's not too late, pop it into the question box and we will get it answered. So the first question is, if I'm doing these arrays, how many clones or organoids can I expect from each array? Allysa or Scott, do you have an answer for us there?
Yeah, sure. That's a great question. So the 500 micron cell raft array has over 2,100 available microwells or positions. Depending on the organoid type, this can vary a little bit, but we expect hundreds of organoids on each array, with a little bit of flexibility depending on the organoid forming efficiency and the particular workflow that you're doing.
Okay, fantastic. All right, we've had another couple of questions come in, so we'll move on to those. How large can the organoids grow on the cell raft array before isolation?
That is another great question. We have successfully isolated organoids up to 1 millimeter in diameter. The organoid size, though, I want to point out, is not restrictive, so to speak, to the growth on the cell raft array. So with all of the organoid types that we have worked with or that our customers are working with, these organoids continue to grow off of the array after isolation. So, if your goal is to isolate organoids and develop them into, fully mature, large organoids of a particular size for your downstream, it's worth noting that you can isolate them from the array and then continue to grow or mature those organoids in the 96 well plate to your desired size.
Okay, fantastic. Thank you very much. We've had another question in here. What is the resolution that you can achieve through the cell raft to image your organoids?
Sure. We have an inverted microscope with a 10x objective. With the camera, we get up to 20x resolution of those organoids, and that includes Z-stack feature as well.
Okay. All right, next question. What type of plates can I isolate organoids into?
You can isolate into any standard SBS 96 well plate format. Here at Cell Microsystems, we've isolated into standard flat bottom, round bottom plates, ultra low attachment plates. The important part is that, when we're calibrating the instrument for your lab's particular use, that we're able to accurately calibrate to the plate type that you are looking to use for your applications.
Okay, thank you very much. Another question. Could you please describe the cellular composition of cerebral organoids? What is the percentage of oligodendrocytes?
That is a great question. So we have not done any single cell sequencing or advanced staining on these organoids. This data is really fresh off the press for us. It is something that we're looking into doing, but we have not yet characterized those mature cerebral organoids to that level.
Okay, fantastic. Thank you. We've got one more question. Is there a risk of cross-contamination during isolation?
No. So the cell raft array is really unique in that each of those cell rafts has an alphanumeric address that the software recognizes. So anytime you're imaging or you're mapping a 96 well plate, the software knows exactly what location that specific cell raft is on the array. So in those, the iPSC workflows that I showed you today, where we had a mixed population, we very clearly mapped plates that had, red organoids and green organoids. And through the process of isolation, which is random based on location on the array, not on location in the 96 well plate, we have 100% accuracy of the cell raft that you've selected going into the particular location on the 96 well plate.
Fantastic, thank you. We just had another question come in. So can replacing different tablets damage the organoids? And if so, how can you avoid it? And will it influence the viability?
Sorry, maybe you can just repeat that question again. I didn't get the very first part.
Sure, of course. So can replacing different sorry, let me start that again. Can replacing different tablets damage the organoids? And if so, how can you avoid it?
I'm not sure what the question is specifically referring to in terms of tablet. Maybe the question is linked to the viability on isolation, perhaps. And we've done a lot of viability staining with organoids on and off the array, and find that the process of isolation into the 96 well plate does not affect the viability or the potential growth of the organoid.
Fantastic. And then the second half of that question is, does the size of the organoid affect the disease that can be studied with it?
I think that may depend on the specific application and organoid type. Of course, there are, some dense, dense organoids where you might be concerned as the organoids get to a larger size that you have less nutrient and gas exchange to the middle of the organoid, and then have some sort of necrotic core. So that's always a consideration when we're building these 3D models. But in terms of that being specific to diseases, I think that would just depend on the organoid or tissue type.
That makes sense. Thank you. Have you found that the persistence of the raft has any effect on biology?
So far, no. We get asked this question a lot. What happens to the cell raft? That is a inert polystyrene microscale growth surface. The organoid, depending on type, sometimes over time and over feedings dissociates from the cell raft. Other times that organoid remains attached. We haven't seen any effect on the biology so far. And because that cell raft is optically transparent, it hasn't given us any lack of access, so to speak, to imaging the organoids post-isolation.
Okay, fantastic. Thank you very much. I don't think we have any more questions coming in there, so I think we will wrap it up there, as that is all we have time for today. So just everyone join me in giving a very big thank you to Dr. Stern and Dr. Magness for coming today and giving that incredibly interesting talk. So just to remind everybody, any questions that that you didn't have a chance to ask, or if you come up with any questions later on, you can still ask those and they will be answered offline as soon as possible. You can even continue to ask questions even when watching the webinar on demand. There will actually be a survey when the webinar ends today, and we'd really appreciate it if you could take a couple of minutes just to give us your feedback on today's event. That would be much appreciated. Thank you very much. A certificate of attendance is available to download from the Handouts tab on the right-hand side of the webinar platform, and the webinar will now be made available on demand. So if you have any friends or colleagues that you think may be interested in this webinar, please feel free to share the link and let them know that they can still ask questions, even when they're watching on demand. Okay, thank you everyone for your time once again and for those extremely interesting talks. Thank you very much.